quite a lot of trouble with this, right? Because you're finding that there are many ways to code for a single amino acid for your growing protein chain. So going back and forth between RNA and DNA was quite easy. There are many examples in biology, both of transcription, and then you can also have RNA going back to DNA, but right, like once you get to the protein being made, there's really no going back, because you'd never know. Some of them you know. So what's methionine? AUG. You always know if you see methionine that the mRNA sequence must have been AUG. But what about serine up here, SER? How many ways are there?
AGU and AGC.
Six.
Four, actually, yeah.
Yeah, so there are 6 triplet codons that could have coded for serine. So once you get to protein, there's no going back. Okay, so let's get the rest of this. Get the rest of this mRNA translated into protein, and then I'll show you in a video, sort of real-time 3D, how this process is occurring. So we've translated our AUG into methionine. We've translated the CAA into proline. We've broken this bond between the tRNA and the methionine. We've made this bond between the methionine and the proline. And then again, it's the ribosome. Maybe I should draw the ribosome in a different color. And it's the ribosome that is doing all of this with the help of these tRNAs bringing in the correct amino acid. So the next triplet up here is UUU. Yeah. The ribosome is a complex that is actually doing the translating. So the tRNA brings in the next amino acid, but it's the ribosome that is making these bonds and breaking those bonds. Otherwise, you'd just be bringing in amino acids and then releasing them.
This whole process,
This whole process is protein synthesis.
right.
So
The ribosome is just this location where all this is happening.
the ribosome is a complex that is doing all of this stuff. So like the RNA polymerase was allowing the complementary base to come in and bond, hydrogen bond, and then be attached to the growing chain. The ribosome is doing sort of the same thing, but because it's not a one-to-one, you have to have the tRNA bring in the amino acid to read in triple.
Okay.
So it's maybe sort of funny to think about, right? It's a protein that's making proteins. Its job is to make
But the ribosome is
proteins.
protein that is complex.
The ribosome is a complex.
Wait, it's not a protein.
I like the
Or is it a complex of proteins?
The ribosome is a complex of many things. Yeah.
You could just, I mean, to simplify thinking about it, you could just think of it as like an enzyme, essentially.
Yeah.
An enzyme that has this super complex active site that can fit all this stuff and catalyze this reaction of breaking the bond between the amino acid and the tRNA and making the bond between the 2 amino acids. Okay. But it's huge, it's massive, and it's lots of RNA and lots of protein and all kinds of stuff.
Okay, so it's really like a summation of all these different parts is the ribosome. Like, because you keep saying complex, but when you say complex, like, there's no specific parts to this.
Yeah.
Like, I don't
There are specific parts to the.
So it's the tRNA and the mRNA and like all these things. Like,
So so the tRNA is the tRNA.
Yes.
The mRNA is this molecule that's being translated.
Right.
And then we'll think of the ribosome as, we'll think of it just as a super protein
Okay.
for right now,
So-
that is able to make bonds, break bonds, and allow these tRNAs to come in and leave and move across the mRNA strand.
Okay. All right.
Okay, so the ribosome.
Is when you're doing with mRNA at the restaurant, that's called ribosome.
Okay, so ribosome is doing a lot of things. It is moving across the mRNA strand. It is allowing the tRNAs to come in with an amino acid. It's allowing bonds to be made between two amino acids. It's allowing the bond holding the amino acid to the tRNA to be broken. And then it's allowing the free now tRNAs to leave. So that's doing a lot
All right.
of things, and I'm simplifying it by saying the ribosome.
Okay.
So it is a complex, but this whole complex together is doing these many jobs.
It'll all be illustrated in the video too, right? I think the video is going to clear it up for people a lot.
The video should help, and if you're really interested in all of the subunit details, there are more there's a video that includes a lot of those. If you go to their website, the DNAlearningCenter.org, there's 2 versions of the video. Okay, let's see. So we are translating our protein. We've got methionine, we've got the proline, so now we have a UUU. So what amino acid are we bringing in? What amino acid? What is UUU? So phenylalanine? So we have, so imagine the ribosome moving along. The video will make this a little better. But now we have, this bond has to be made between our proline and phenylalanine. That bond broken. And then what about GCA? Hmm?
I'm sorry. Alanine.
Alanine? So now bond made, bond broken, and then what about UAA?
Wait, no, that's, am I reading this right? Is that like a stop?
Stop. So the protein has to stop somewhere, right? So there are, same as there are triplet codons that code for these amino acids, you code for termination the exact same way. So this, yeah.
This is gonna, if I'm right, this will just clear everything up for me.
Okay.
It's building a protein.
We are building a protein.
So that thing is a protein.
This is, yeah, this is our string of amino acids, or polypeptide chain. That is our protein.
Wonderful, beautiful. Okay.
So for clarification on the worksheet,
Oh.
is there a specific reason why the chart on the front that gives us all our amino acid translations uses the nitrogenous bases of DNA, specifically thymine, whereas the one on the second page uses uracil?
Yeah, so you remember when I had the DNA drawn?
Yeah.
So there's our DNA sequence, and then what's our mRNA sequence?